Hey, folks, what's going on? It's Dr. Kaz. I'm bringing you another review session. This is the second part of Chapter 4. I have to confess something to you before we get started. I actually made this recording yesterday, and to be honest with you, I thought it was an awesome recording, but the whole time I had the audio off. So here I am sitting down doing this again. So I'm only hoping that this will be better than the previous one. So that being said, let me jump right in and talk about cytology. You know, last video we talked about the three parts of the cell. Okay, the cytoplasm, the plasma membrane, and the nucleus, and we finished the cytoplasm. Now we're going to move on to the plasma membrane. All right, a couple interesting facts about the plasma membrane. It's made up of proteins and lipids, and they are both equal in weight. Even though there's numerous phospholipids, much more than there are proteins, it's by weight they are equal. Okay, an important concept that you need to know that we will be talking about a lot in this video is that the plasma membrane regulates the movement of substances across the plasma membrane or in or out of the cell. That is very important that you know that. Now, certain things can get by through the plasma membrane with no problem at all. They don't need any help. That means they can cross without assistance. They have to be small, and they have to be nonpolar. Those two attributes will allow substances to move right through the plasma membrane without any help whatsoever. So, that being said, the plasma membrane is made up of three types of lipids. The most numerous is the phospholipids, cholesterol, and then glycolipids. All these we've talked about in Chapter 2. Okay, so now we're going to get into a little bit more detail with that. If you look at this next picture here, I'm going to zoom in here for you folks, just to kind of give you a little bit of an idea of what we're looking at here. All right, so you can see here, here's our phospholipid bilayer. All right, so you have one layer of phospholipids. The head is facing out. This is the extracellular space or the interstitial fluid area here. Okay, that's outside the cell. This is inside the cell. So what we did is we took two layers of phospholipids and we put them back to back. So the polar heads, which is the hydrophilic portion of the molecule, is facing out. And then on the inside of the cell, we have the polar heads facing in. Again, hydrophilic, water-loving, because there's a high water content both outside the cell here and inside the cell here. The hydrophobic area, where the tails are, that's on the inside here. And you'll notice on the inside, you'll also see parts of the cholesterol. Cholesterol helps the cell plasma membrane to withstand. It actually strengthens the plasma membrane. It helps it to withstand extreme temperature changes. And then on the outside, we also have, all right, these glycolipids. And these glycolipids are just a carbohydrate group attached to a lipid group, all right? And it helps form what we call the glycocalyx, which is the sugar coating of a cell. Okay. All right. One other thing I want to point out is that there's proteins in the cell. And so you can see the plasma membrane has, there's a numerous amount of phospholipids compared to the proteins. But the proteins, there's two types. You've got this one here that spans the entire width of the plasma membrane, and that's called an integral protein. And then you've got a peripheral protein that either sits on the inside of the plasma membrane or on the outside, floats like trash on a body of water, okay? And that's what those peripheral proteins do. And proteins have several functions. Actually, a lot of the functions of the plasma membrane has to do with the proteins, okay? So like we said before, half of the plasma membrane weight is going to be made up of these membrane proteins, all right? So they're going to float along inside of that phospholipid bilayer there. And most of the membrane's functions and duties and responsibilities are controlled or relegated by these membrane proteins, okay? So there's two types. Like I mentioned in that slide before, there's the integral and there's the peripheral, okay? So back in Chapter 2, we talked a lot about the different functions of proteins. Now we're going to go and kind of review those functions, but also mention, all right, a little bit more detail about some of these functions. So a big function for these proteins is that they're involved in movement across the plasma membrane. And they're involved with regulation of movement of certain substances that it can move into the cell and those that can move out of the cell. So we'll talk about some of these here in a few moments. But some of the examples of these transport proteins are channels, carriers, pumps. We'll spend a lot of time in later chapters talking about these three, and then we will discuss the symporters and the antiporters here, but we don't really cover that too much in this course. All right, the next function of a membrane protein are cell surface receptors. I want you to think of a lock and a key when we talk about cell surface receptors. All right. So you have a lock, all right, and in order to open that lock, you need the key. So another name for a key when we're talking about cell surface receptors are ligands. And those are specific molecules, chemicals, all right, that your body has. All right, for example, we'll talk about the neurotransmitters here that are used with cells. All right, neurotransmitter could be acetylcholine, it could be a hormone. Regardless, it floats around. And it has a special configuration, and it floats around and it looks for a receptor that can match that configuration. And that's how neurons can communicate to one another, all right? But also how neurons can communicate with their what we call effector organs, like muscle, okay? And when it affects a muscle, it can initiate a contraction, okay? So think of the cell surface receptors as a lock that's locked until we get the right key, which is going to be a specific type of molecule, all right, for example, neurotransmitter that can open it up and actually activate it. The next type of function is the identity markers. It's very important that your white blood cells, your immune system, know which cells are supposed to be a part of your body and which cells are not supposed to be a part of your body. And these identity markers help with that. Because if you have a cell that's in your body that's not supposed to be there, it can be harmful, and you need to eradicate that quickly. Well, these identity markers which are on your cells that identify your cells as being a part of you, okay, which identifies these cells as what we call self cells, okay? These identity markers play a huge role in that. And sometimes if these identity markers kind of become down-regulated or destroyed because maybe a cell gets too old, that's no problem. Because then, all right, a white blood cell can come along and destroy the cell and get rid of it, so you can either have a new one, or if it's a foreign invader, a cell that's considered to be non-self, like a bacteria, right, or a cancerous cell, then we want to destroy those cells too. Enzymes are an important functional category for membrane proteins because cells undergo a lot, and when I say a lot, of chemical metabolic processes, okay? So in order to start some of these chemical reactions, much like you need to start a fire, right, you need a flame. A lighter, for example, is a catalyst to a campfire, let's say. Well, enzymes are a catalyst, all right, to start these chemical reactions that a cell might need in order to synthesize something. All right, or to metabolize something, that if it takes in some nutrients from outside the cell and it brings it into the cell and it needs it for energy, let's say, to make ATP, well, you need enzymes to help catalyze those chemical reactions. So these enzymes can be on the inside of the cell or they can be on the outside of the cell, okay? These last two, anchoring sites and cell adhesion proteins, all right, that has to do with cell stability and cell structure. The anchoring proteins, all right, are just going to be these sites, all right, they're usually peripheral proteins, some can be integral, that the cytoskeleton, the cell cytoskeleton will attach to. And it helps to stabilize cells and give them their shape, maintain their integrity. It's very important. The cell starts to lose its integrity and its stability, then it can degenerate and become damaged, and then it has to go. All right, cell adhesion proteins, we see this when we're looking at the epithelial cells. Epithelial cells are very tightly packed, okay? And that has to do with, all right, these cell adhesion proteins that attach these cells very close to one another, so things can't slip in between the cells unless the cells, that type of tissue is made for that, okay? So this next slide, you're going to see some pictures here of that. All right, let me zoom in here. All right, so here you can see a channel. All right, so we have something on the outside of the cell. We need to get inside the cell, so these proteins allow for a channel to be created. It's usually water filled, and then a certain substance can move across the plasma membrane. Carrier protein is also helpful. For example, glucose, if we got to get it inside the cell, all right, this actual carrier protein will change its configuration, it'll change its shape, and it'll move something from outside the cell to inside the cell, and vice versa. Pumps are what we use to move substances against their concentration gradient. We'll talk more about that, all right? But it is an energy requiring structure, and again, it'll move things across that plasma membrane. Our cell surface receptor, this is our lock and key mechanism. Here's our lock, here's the specific key, that ligand. And once it activates the receptor, it can initiate some sort of process to occur. It could be a chemical process, it could be an immune reaction, it could be whatever. All right, but the cell surface receptor is important. Identity markers here, this is what allows us to identify what's supposed to be, where it's supposed to be. Here's our enzyme configuration here. You have a substrate, that's the ingredients for a recipe. All right, and then the enzyme is like the oven. It kind of cooks it and causes the chemical reaction to move along, and then you have your finished product. So it's like baking brownies. You need eggs, all right? You need the brownie mix, all right? Some oil or whatever, all right? And that's your substrate. And then the finished product is your brownies. All right, and the last two is what I was telling you about. Here's the anchoring site and our cell adhesion proteins here that allow for cells to maintain their structure and integrity, okay? And then how cells can be tightly packed and close to one another. All right, so we'll talk more in detail on some of these things. All right, now after this chapter, you're going to really have a decent appreciation for the plasma membrane of a cell. Remember, your cells are the functional units of your body. So for a lot of these physiological processes to occur in your body, it breaks it down to the cellular level, but also to the chemical level, the interaction of certain molecules and ions and whatnot. Well, the plasma membrane plays a big role in this. So this is a very important fact here, and I'm going to circle this here for you. You have to understand that the plasma membrane is going to regulate movement of substances in and out of the cell. It's huge. At the same time, it acts as a physical barrier. It's a limiting barrier. So it limits what can move across into the cell and what can move across out of the cell. And that's a huge concept to understand. So it just doesn't allow anything to cross over at its leisure. It regulates what's moving in and out. And later on, when we get to chapter 12, we'll talk about this important concept, the electrochemical gradient. Certain chemicals, when they move across the plasma membrane, all right, they can change the actual the electrical charge, because some cells will have an electrical charge component to it, namely neurons, all right? And with the movement of these chemicals, it can change that charge, which is important because a nerve signal, all right, is essentially an electrical signal moving down the neuron. More on that in chapter 12. And then also, if cells need to communicate with one another, or if they need to communicate within themselves, the plasma membrane plays a big role with that. So there's two types of membrane transport processes, and we break them down into two categories, passive and active. Okay, passive requires no energy, active does require energy. Okay, so passive and active. So on this next slide here, you'll see, all right, this figure 4.7. All right, this is in chapter 4. Again, to break this material down, I always like to start with the easier or the more simple material, okay, and then get into the more detailed stuff. So this is what I would recommend for you folks when you're first learning this, because this is a lot. So I would start to learn and memorize whatever's in these boxes here. And then when you feel pretty confident with that, then you can start to move on to more of the detail. Now remember, this slideshow, this recording is a review recording. At this point, I'm assuming that you've gone over this material already and that you're using this recording to review that material, okay? And that's how I'm teaching it. So I would say start with these individual boxes, learn what's in each box, then you can move on. Okay, so we're going to start off here, all right, with the passive processes, then we're going to move over to the active processes. So we're going to start here with diffusion. And as you can see, there's two different types of diffusion. So we're going to move into that. So first, you have to understand what diffusion is, okay? And it's essentially moving, all right, a substance from an area of greater concentration to an area of lesser concentration. So I'm going to draw this diagram here. It's like a ramp, okay? And that ramp represents what we call the concentration gradient, okay? So at the top of the ramp, we'll have 99 molecules of something. And at the bottom of the ramp, we'll have one molecule of that same something. So nature loves balance. It loves the concept of called equilibrium. And you've heard this term quite a bit in the news over the past few months. We want to flatten the curve. Well, that's what we want to do here. We want to flatten this sloping here, this curve. So if I have a boulder at the top here and I'm going to push it down a hill, it's going to roll down the hill. It's going to go down that grade or that gradient. So all of those molecules, the 99 molecules here, all right, and you have one molecule down here, that's a 98, because if you subtract 1 from 99, that's 98. It has a difference of 98. That's a large difference. Ideally, with nature, it wants to balance everything out. It wants it to be in equilibrium, so it wants to have 50 molecules of whatever on one side and 50 on the other. All right, when we get that, when we get equal numbers there, we call that equilibrium. Okay, so diffusion will occur until we reach this equilibrium, balanced, 50-50. Okay, and basically that means where everything is kind of spread out, and I'll show you what I mean by that. So first of all, know this. All right, the net movement of a substance is going to go from an area of greater concentration to an area of lesser concentration. All right, so the 99 molecules up here and the one molecule down here, they're going to want to move down the concentration gradient. Down the concentration gradient. So a lot of molecules, and we're going to include ions in that, and that means sodium, potassium, calcium, chloride, all those important molecules and ions that we need to know for this course. All right, they're moving around. And they're moving around because of kinetic energy. Kinetic energy is the energy that generates movement. All right? So when things are moving around, it's because of kinetic energy. But at the same time, as certain structures are moving around, they too are generating kinetic energy. So we're going to have these things move around until we establish equilibrium. And what equilibrium is going to also mean, and this is important that you know that, it's when the molecules are evenly distributed through a given area. So I'll show you an example. Diffusion. We have a medicine dropper here with food coloring, and we have a beaker full of water. So we take that food coloring, we dump it in the water. We notice right here, this is an area of greater concentration of that food coloring. All right? And outside here is an area of lesser concentration. Okay? So what's going to happen with the process of diffusion? Okay? We want to go from an area of higher concentration to an area of lower concentration. So these molecules of the food coloring are going to start to spread themselves out more and more and more until we get equal distribution throughout the entire beaker. Okay? And because of that movement there, it's moving down its concentration gradient, all right, until it's established equilibrium. And that's diffusion for you. So the same thing is going to occur in your plasma membrane. Okay, if you have a substance, all right, here's your cell, all right, and say you got sodium, for example. You got about 140, well, let me keep with the normal numbers here. You got 99 molecules of sodium outside the cell and 1 molecule of sodium inside the cell. Knowing what we know, with diffusion, a substance wants to move down its concentration gradient. It wants to go from an area of more to an area of less. Which direction is sodium, in this example, going to move? Is it going to move from outside the cell to inside the cell? Or is it going to move from inside the cell to outside the cell? I'm going to give you a second to think about that as I take a sip of my coffee. If you said that it is going to move from outside the cell to inside the cell, you are correct. It's going to move down its concentration gradient. Okay? All right. So now that we have a decent understanding of diffusion, let's talk about a specific type of diffusion called simple diffusion. Now we're going to move into our cell physiology here. So, simple diffusion is quite easily understood as when molecules pass across the plasma membrane. Okay? And in fact, remember what we said, that phospholipid bilayer is a limiting barrier. But, all right, but if you have a molecule that's small, or if that molecule is nonpolar, it can slide in between those phospholipid molecules, no problem. No problem. Okay, so they have to have, you want to make sure that you know this. If the molecule is small, wow. If the molecule is small, all right, and nonpolar, it will move right between those phospholipid molecules. So that includes your respiratory gases, oxygen, carbon dioxide, they move right through the plasma membrane. Some smaller fatty acids, alcohol, and urea, which is a waste product of the cell. Like urine is your waste product, you know, from whatever you're eating and drinking. All right, well, urea is the cell's waste product, and it's got to get rid of it. Well, it's nice that it's not being regulated by the plasma membrane, and that's true. With simple diffusion, the plasma membrane does not, make a note of this, does not regulate the movement, all right, of these molecules. But what does regulate the movement of these molecules is the concentration gradient. Okay, if you have more on either side of the plasma membrane, it's going to move from an area of higher concentration to lower concentration. And as long as that gradient exists, you will have movement. All right, until we flatten the curve, so to speak, you will have movement. So let's look at this picture here to better explain this. Okay, you can see we've got our plasma membrane here. This is the outside of the cell, which is called the interstitial fluid, or it's also called the extracellular space. Okay, outside the cell. Wow. Okay, and then the inside of the cell, the cytosol. So you notice that we have many more molecules of oxygen outside than inside. So oxygen is going to want to go down its concentration gradient. It's small and it's nonpolar, so it slides right through the plasma membrane and enters into the cell. Same here with carbon dioxide. We have more carbon dioxide inside the cell than outside. So it too will go down its concentration gradient, and it'll move from inside the cell to outside of the cell. Okay? Until equilibrium is established. Important that you know that, folks. If you don't, pause the video, listen to it again. If you still don't understand it, go back into the chapter, all right, and read that material. The second type of diffusion is called facilitated diffusion. When you facilitate something, you're giving it assistance. You're giving it help, okay? So with facilitated diffusion, all right, what we have is a molecule, all right, that is small, but it's also charged, like sodium, potassium. Those are ions. They're small, but they're charged, okay? Or they're small but polar, so they need help. They can't slide in between the phospholipids. So where do they go for help? All right, they're going to go to the plasma membrane proteins, because as you recall, one of the functions for a plasma membrane protein is transport. So they're going to utilize these proteins for help, and so it'll help with transport. Okay, so there's two types. We have the channel-mediated diffusion and carrier-mediated diffusion. Okay, channel-mediated diffusion, carrier-mediated diffusion. All right, so let's go into a little bit more detail about that. The first one for channel-mediated diffusion is, remember I was talking about this with the transport proteins, you'll have these channels, all right, and they'll have this It's kind of like this opening that they established with the plasma membrane, right? And it's filled with water, but these channels allow for the movement of these small charged particles, ions, so sodium, for example. The nice thing about these channels is that they're only going to allow the movement of one specific type of ion. So what that means is sodium can only go into sodium channels. They cannot enter into the cell or leave the cell through a potassium channel. So it is ion specific. Okay? Ion specific. So there's two types of channel mediated, or there's two types of channels. The first one is the leak channel. They are always open, like 7-Eleven, open 24/7. They don't close. So stuff is always going to move through those channels. And when I say stuff, one specific type of stuff. So sodium leak channels are only allowing sodium to move through those channels. And they'll only move through those channels if a concentration gradient occurs. Okay, it has to be present. All right, the other type of channel is a gated channel. So if you have a gate in your backyard and your dogs are out in the backyard, they'll stay in the backyard if the gate's closed. Okay, if you open up the gate, they'll leave. Very similar when we're talking about the ions. These types of channels are closed normally, and the only thing that will open them up is some sort of stimulus. And that stimulus could be a certain chemical being present. All right? So if that chemical is present, all right, it can open up that gate, and then it allows the ions to move through the gate. All right? And another type of stimulus could be an electrical charge. That too can work. Okay? All right. So you have to understand that these channels, leak channels and gated channels, we'll get into more detail about them. So I just kind of want to give you an introduction, but we'll get into more detail about them when we get into chapters 10, which is muscle tissue, and chapter 12, which is the nerve tissue. Okay? So just understand these basic characteristics here. And then we'll talk a little bit more in detail in those latter chapters. Okay. The other type of facilitated diffusion is the carrier-mediated diffusion. So what we need is we need help, again, to get a certain type of molecule across the plasma membrane. And again, small polar molecules, right, need help. So we have this special kind of protein, we call it a carrier protein. And what it's going to do is it's going to change its shape as that substance is entering and moving across the plasma membrane. And this shape change, all right, allows us to see or see how that molecule is facilitated across the plasma membrane. Okay? So again, these substances are going to move down their concentration gradient, moving from an area of higher concentration to lower concentration. And so when this substance or this carrier moves only one substance, we call it a uniporter. Uni meaning one. Okay? So if you have one carrier protein in the plasma membrane compared to 10 carrier proteins, you're not going to have as much of that substance moving across the plasma membrane. So the more carrier proteins you have, the more of that substance can move across the plasma membrane. And this is important when we get into for glucose molecules and whatnot, all right, and how they're able to enter into some of the cells and certain hormones. So the number of channels and carriers is going to determine the rate, how fast of a substance can move across the plasma membrane. So keep in mind, if you have more channels and more carriers, right, you'll get an increased rate. If you have very few to none, then you have a decreased or low rate. Okay. So here's a picture of that carrier protein. So here's your glucose outside the cell. We need it inside the cell because we need to make some energy. So it'll move across into this carrier protein here. It changes its shape, as you can see, and then it spits it out onto the inside of the cell. Okay. Now it could also do it moving things on the other side, vice versa. All right. Not just for protein. I mean, not just for glucose. All right. That's the first passive process that we talked about. All right. So that is the first. Diffusion is the first of two passive processes. The second one is osmosis. When you see osmosis, all right, immediately, first thing that should come to your mind is water. Osmosis is the movement of water. In diffusion, we were talking about the movement of a whole bunch of different things. Okay? But with osmosis, we're talking about the movement of water. So what's a solute? Everything else that's not water. Sodium, glucose, all right? Fatty acids, whatever, urea. All right, that's what we're going to consider for our purposes to be a solute. Okay, so osmosis is the movement of water. And two, with osmosis, the water has to move through a selectively permeable membrane. For our purposes, guess what our selectively permeable membrane is going to be? You guessed it, the plasma membrane. Okay, plasma membrane is our selectively permeable membrane. So the plasma membrane is going to allow water to move right across, but it's going to prevent the movement of the solutes, the other stuff. Okay? So because of this principle, we're going to see a difference in water concentration on each side of our plasma membrane, which means you'll see more on one side and less on the other. It could be more on the inside and less on the outside, or vice versa. Okay? Okay, so the only thing when we're talking about osmosis is the movement of water. Now, this next slide here is a very important slide. So I'm going to let you start to take a peek at it and read some of the information while I take my sip of water here. That's a lie. That was a sip of coffee. All right. So a couple things we're going to talk about. One is the concentration gradient, and the other is the movement of water. Okay? So we have to establish a concentration gradient, all right, with our cell. So that means we're going to be discussing the inside of the cell, which is the cytosol, and then the outside of the cell, which is our interstitial fluid. So knowing what we know with osmosis, only water can move. So the solutes cannot, they are prevented, they cannot move across the plasma membrane. Okay? And because of that, all right, you will see that you'll have more water on one side of the plasma membrane and less on the other. So that's going to create a water concentration gradient. Okay? Because the solutes can't move, but water can. So this next part is important. So I'm going to read it out to you. Greater, you need to just memorize this. Okay? And hopefully over the next couple of slides, you'll understand it. Okay? But for right now, memorize it. Greater concentration of solutes is going to contain a lower concentration of water. Okay? Keep that in mind. A greater concentration of solutes is going to have a lower concentration of water. So, to give you an example, you've got 99 molecules, and let's just say sodium on the outside of the cell, and one on the inside, like we used before. Okay? So, I've got more sodium outside the cell than inside the cell. I have a greater concentration of sodium outside the cell than inside the cell. So going back to this statement here, greater concentration of solutes contains a lower concentration of water. So what that means now, okay, sorry folks, I was trying to see if I could change the color. Oh, I can. All right, give me a second. Beep, beep. So what that means now is I am going to have, according to this, all right, when we're talking about the solute concentration and the concentration of water, what that means now is I'm going to have 99 molecules of water. I'm just going to put H here, okay, for H2O. And 99 molecules of water on the inside and 1 molecule of water on the outside. Again, this is just for our example here. All right, so again, I'm going to kind of reiterate what I'm saying here. Greater concentration of solutes contains a lower concentration of water. So let's establish our solutes first. 99 molecules of sodium on the outside, 1 molecule of sodium on the inside. So by that rationale, that means, okay, where we have the greater concentration of solutes, we're going to have the smaller concentration of water. And vice versa, where we have the smaller or lower concentration of solutes, we'll have the higher concentration of water. All right? I want you to kind of understand that as best as you can right now. Don't worry, I'm going to keep going in and kind of hammer this home more, all right, over the next few minutes here on this slideshow. Okay? So just for right now, we're going to use this example here, okay? And we're going to go with that. So, knowing what we know about osmosis, all right, water is going to move from an area of high water concentration to low water concentration. So here's our high water concentration, and here's our low water concentration. So, will water move from inside the cell to outside the cell, or will it move from outside the cell to inside the cell? Mind you, osmosis is a passive process. So when we're talking about water, because it's osmosis, it's the movement of water, and water is always going to move down its concentration gradient. So I'm going to give you a second to figure out what your answer is going to be. Does it move from inside to outside the cell, or does it move from outside the cell to inside the cell? If you answered that it moves from inside the cell to outside the cell, you are correct, because water moves down its concentration gradient. 99 molecules inside the cell, 1 molecule outside the cell, it will leave the cell. So take a look though at what we just covered here. If water goes from an area of high water concentration to an area of low water concentration, look at the solutes. You have low concentration inside the cell versus high concentration outside the cell. Could we not also say that water moves from an area, all right, of low, no, let me rephrase that. Could we not say that water will also move into an area of high solute concentration? Check it out. Remember, it's moving down its water concentration gradient, but at the same time, it's moving from an area of low solute concentration to an area of high solute concentration. So we can say that water is going to move into the area where there's greater solute concentration. This statement here refers to the concentration gradient across the plasma membrane. This is before any water moves. All right, so this is prior to the movement of water. So the greater the concentration of solutes contains the lower concentration of water. All right, so then when water starts to move, it's going to move down its concentration gradient, obviously until equilibrium occurs. We can see in this case here that it's going to move. Now the example here is 1% solutes and 3% solutes. We can change that and scratch that out and say, instead of 3% solutes, our example was 99. Regardless, doesn't matter, okay? But what we can say is that water is going to move from a solution, all right, which is the inside of the cell, the cytosol, of that 1% solutes, which has that higher water concentration, into the solution, which is that space outside the cell, where we saw 99 solutes, the lower water concentration. See? Lower water concentration outside the cell, but the higher solute concentration. If this is a lot to take in, pause this video, step back for a moment, rewind it, and then sit back down. Okay? That's implying that you stood up. All right? So again, if it's still not making sense, no problem. Rewind this video at the beginning of osmosis and listen to it again. Okay? This is an important concept that you need to know, because we're going to get into something here quite important here when we get into the osmotic pressure and the concept of tonicity. So let me show you a picture here. You can see here, all right, zoom in a little bit. You can see here, the little red dots here are our water molecules, okay? And you can see that there's greater concentration outside the cell than inside the cell, and water will move right across the plasma membrane here. And it also has these structures called aquaporins, and they're just protein channels, okay? And they let water just seep across the plasma membrane without having to go through the plasma membrane. So that helps the movement of water even more. But you can also see how the plasma membrane is rejecting certain solutes. All right, this one here could be a charged ion, for example. Glucose is too big, so it can't make it through. Okay? So it's non-permeable to most solutes because of it maybe being charged like an ion. It could be polar or it could be too large. Remember, the only thing, all right, I shouldn't say the only thing, but what allows something to cross through the plasma membrane, it has to be small and non-polar. Then they can make their way through. Okay? All right, so osmotic pressure. I look at osmotic pressure similar to your faucet, your kitchen faucet. When you wash the dishes, all right, do you let the water just trickle out of the faucet, or do you turn it up a little bit to blast some of the food particles off? Well, I'm assuming you're like me and you turn it up a little bit to blast the food particles off. That's water pressure. Osmotic pressure is similar to that. So when you get that water moving, when it starts to move from that area of high concentration to low concentration, it creates a pressure, all right? As it's moving across our plasma membrane. I'm going to change the color back to blue. Okay? So please keep that in mind. When water moves, it starts to exert, all right, some pressure through that movement of high concentration to low concentration. All right, so when we get that water moving across that semi-permeable membrane, aka the plasma membrane, it exerts a pressure. Okay? And at the same time, all right, remember what we said. When water moves from an area of high water concentration to an area of low concentration, it also is moving towards the area of greater solute concentration. All right? So that's what we're seeing here. We have a difference in solute concentration, and so that is going to, and so therefore you have a difference in water concentration. And so that is where we're going to see that pressure being generated. So just like that example I told you about rolling a boulder down a hill, okay? The steeper the hill, well, let's not even use that. Let's talk about a bicycle. You're on your bicycle, you're riding along. If you go to the top of a steep hill and just coast down, compared to if you go to a very a gradual kind of incline, all right, or decline, I should say, you're going to move down the steeper hill compared to the less steep hill. And that's the same thing when we're talking about osmotic pressure. if we have a steeper gradient, that example, like I was saying, 99 molecules on one side and 1 molecule on the other versus 55 molecules of water on one side and 45 molecules on the other, the steeper that gradient, the bigger the difference, okay? The more water is going to move because we generate a greater osmotic pressure. So this will be 99. Come on now. Oh, Lordy, come on. Versus 1, and this one over here is 55, okay, versus 45. So it's much less steep, so the pressure will be less on this bottom one compared to the top one. Okay? So a term that you want to get familiar with is hydrostatic pressure. This is the amount of pressure inside your water pipes in your house. It's also the same concept applies to your blood vessels. When your blood is flowing through your blood vessels and it's moving down, it's always pushing on the inside of the blood vessel wall, trying to get out, trying to push out. Okay? So hydrostatic pressure is that pressure exerted by a fluid on the inside wall of whatever its container of whatever we're measuring. So if it's the pipes in your house or your actual veins, arteries, capillaries in your body. Okay? That's that outward pushing pressure. All right? So when we talk about osmotic pressure, let's use an experiment here. So here we have this U-shaped tube, and on the side B here, our right side, if you count it up, you'll notice these little red dots, those are your water molecules. We have more water molecules on side B than we do on side A. Okay? In the middle, we have a semi-permeable membrane, and that's only going to let water move. These bigger black molecules here, those are our solutes. So you notice there's 4 on side B and there's 8 on side A. So we can say we have, all right, low solute concentration here compared to the high solute concentration here on side A. Also, then we can say, too, we have a higher water concentration on side B compared to a lower water concentration on side A. So pause it if you need to. Take a look, okay? So this is prior to the movement of water. Okay, so knowing what we know, that water likes to move from an area of high water concentration to low water concentration, and it also likes to move toward the area of higher solute concentration. Which way is water going to move in our experiment? Is it going to move from side A into side B, or is it going to move from side B into side A? Take a second. Let me sip my coffee. Write down your answer. If you guessed that it's going to move from side B into side A, you are correct. That's what will happen because it has the higher solute concentration and the lower water concentration. So it's going to move until it's equilibrium. Please, please, please understand that. So that leads me to my next concept. And that has to do with what we call tonicity, which is the ability of a solution to change the volume or pressure, which is what we refer to as tone, all right, of a cell by osmosis. So we're going to set up an experiment here in a moment in which we take a red blood cell and we drop it into 3 different types of solutions, okay? And more on that in a second. But when we talk about a cell can either gain water or lose water through the process of osmosis, all right? And when it does that, it will affect the cell's volume, how big or small it is, and it will also affect its osmotic pressure, the pressure inside the cell, all right? So this is, and when we talk about tonicity, this is what we're referring to, how a solution, whatever that cell is in, is going to affect, all right, the osmotic pressure of that cell. So there's 3 types of solutions. Isotonic, iso meaning it's the same. Hypo, you know, is less. And hyper means more. So let me give you the 3 examples here. The first example is an isotonic solution. So we take our red blood cell, we put it into an isotonic solution. What that means is the solution has the same exact number of solutes, all right, outside that cell than inside the cell. So knowing that, if you have the same concentration of solutes both inside and outside the cell, and we know that water always wants to move towards the area of greater solute concentration, will we get movement of water? And if we do, where will it go? If your answer is we're not going to have any movement of water, you're correct. Because you have the same number of solutes inside and outside. If you have the same number of solutes both inside and outside the cell, then water will have the same concentration gradient inside and outside the cell. Because remember, if you have a higher concentration, a solute concentration, you'll have a lower water concentration in that same area. But if it's the same, then everything is balanced and nothing moves. Now we take a red blood cell and we put it into what we call a hypotonic solution. And what that means is the solution has less solutes in it, meaning that there's fewer solutes outside the cell than inside the cell. So water is always going to want to move to the area of greater solute concentration because that's where there is less water. So it's going to move from outside the cell to inside the cell. The cell will start to swell up and it will burst. It's called lysis. Specifically, we're talking about red blood cells or erythrocytes. It's referred to as hemolysis. And the final type of solution is hypertonic. So this is when the solution has a greater concentration of solutes outside the cell than inside the cell. So the water is going to move from an area of low solute concentration into an area of high solute concentration. So it leaves the cell shrivels up, and that process is called crenation. It shrivels up. All right, that's it for passive processes. Trust me, for the active processes, it won't be as in-depth and detailed. All right, those passive processes you need to know, you're going to use them in several later chapters in this semester, and guess what? You're going to see it all again in 211. So learn it now, otherwise you'll be struggling. Okay, so active I can't talk today. Active transport processes, okay? They require energy. All right, so some of these processes we're going to talk about are active transport, but also vesicular transport. We're going to use a bubble, a vesicle, like a car, all right, to move stuff around. So in this situation, active transport, we learned with passive transport that we move things down the concentration gradient. Well, with active transport, we are now going to move things up the concentration gradient or against the concentration gradient. Same type of situation, 99 at the top, 1 at the bottom. Okay? It's like rolling a boulder up a hill. It takes a lot more energy to roll a boulder up a hill. Once you roll a boulder, you push a boulder and it starts to roll down the hill, you don't have to exert any more energy. Okay? So in this situation, you have a higher concentration in one area and a lower concentration in another. Well, we're going to now start to make that area of higher concentration even more higher. Okay? All right. So when we talk about the energy source for these active processes, we'll have a primary energy source and a secondary energy source. More on that in a few moments, okay? But we're still going to be discussing, since we're dealing with the plasma membrane in the cell, we're still going to be discussing about that concentration gradient, all right, across that plasma membrane. So the interstitial fluid outside versus the cell inside. Okay? All right, so keep in mind, against the concentration gradient. or up the concentration gradient. So the first type of active processes that we're going to talk about is the ion pumps. And they're a primary energy source, meaning, all right, in order to use these pumps, we have to use ATP, and that ATP will directly affect the pump and the movement of whatever solutes it's going to move. So the first example that we're using is the calcium pumps that we find in erythrocytes, or red blood cells. And it tries to pump calcium out of the cell. Okay? Although calcium wants to enter into the cell because there's a lower concentration of calcium in the cell than outside the cell. But the cell needs to maintain low calcium inside. Because if you start to increase calcium on the inside of the cell, the plasma membrane becomes more rigid. And that's not good. A rigid plasma membrane of a red blood cell is bad because red blood cells have to squeeze in and out of things all the time. And if they have a rigid plasma membrane, they'll break apart and they'll burst. We can't have that. So we need to keep its plasma membrane nice and fluid. Okay? So we have these calcium pumps to do that. The other type of pump is the sodium-potassium ion pump. This is a very important pump because we're going to talk about it in much more detail in Chapter 12. We talk about the neurons. All right, so you can see that it's a specific pump, which means it's sodium and potassium specific. So it can only pump those two ions. It can't be pumping chloride, all right, or phosphorus. It just doesn't do that. And it's what we refer to as an exchange pump. So what that means is for every 3 molecules of sodium that we pump out of the cell, we're going to pump 2 potassium molecules into the cell. And it's only going to cost us 1 ATP. Okay? So it's an exchange pump. But both of these substances are moving against their concentration gradient. There's more sodium out of the cell than there is inside the cell. So sodium wants to move from inside the cell, sodium wants to move from outside the cell to inside the cell. Well, this pump keeps pumping more sodium out of the cell. So it's going against its concentration gradient. It's an important concept to know. All right, the secondary active transport. This, all right, has to do with where we get energy, all right, to move one of the substances. So one substance is always going to move up its concentration gradient. All right, always up. And remember, we talked about kinetic energy. Kinetic energy is the energy that moves things around, things that are in motion. Okay? But also, when something's in motion, when it's generated, it can also generate some energy. Okay? So as one substance is being moved up its concentration gradient, and that's where we're using the ATP, we can also move another substance down its concentration gradient from the energy that we got from that first substance that was moving up its concentration gradient. Okay, so we're kind of cashing in on some energy that was generated from the first substance. All right, so as we're moving substance A up its concentration gradient, that movement has generated enough kinetic energy where we can move a second substance down its concentration gradient. So there's 2 types of that we can of movement, this active transport that we can use. One is symport, the other one's antiport. Symport is when both of those substances that we're moving are going to move in the same direction. And antiport is when we're moving both those substances, they move in opposite directions. But it always starts with the first substance. Substance A is going to be moved up its concentration gradient. And so the energy that it's generating from its movement going up its concentration gradient, a second substance can move down its concentration gradient. But it can move either in the same direction as the first substance, or it can move in the opposite direction of the first substance. Okay? All right. Another type of active transport is this process of exocytosis. We talked briefly about the Golgi apparatus. Remember, it's the warehouse of the cell. It's going to package up things and modify proteins and whatnot. And if it's going to modify and package up a protein, it can do two things with it. It can send it to the plasma membrane, in which the contents will be incorporated into the plasma membrane, or it can export, all right, those proteins out of the cell. Well, it needs a way to get those packages to the plasma membrane. Okay, much like you need a way to get packages to your house. All right, you have a delivery vehicle, UPS, FedEx, whatever, will bring it to your house. All right, well, your cell has come up with its own delivery vehicle, and that's what we call a secretory vesicle. So you've got the secretory vesicle filled up with some proteins that it wants to send out of the cell, and so it sends it up to the plasma membrane. It merges with the plasma membrane, and as it's merging and opening up, it will eventually expel its contents to the outside of the cell into the interstitial fluid. That process is known as exocytosis. It's exiting the cell. EX, exit, EXIT, okay? That's exocytosis. Another active process, all right, is called endocytosis. This is the opposite of exocytosis. So this is when there are materials outside the cell that the cell wants to take in. Right? It's going to enter, enter. Okay? So it's very similar to exocytosis, but it is in reverse. Okay? So again, if your cell is looking for some nutrients, maybe, it will take up those materials for digestive processes. Okay? So there's a whole bunch of different roles that this plays when we're dealing with endocytosis. Okay? So there's three types of endocytosis. Let's list them off. Phagocytosis we saw earlier. That's the Pac-Man cell, like the macrophage. It comes around and it gobbles things up. Okay? That's phagocytosis. Penocytosis is what we refer to as cell drinking. If there's fluids outside the cell, it will absorb them in. And then there's receptor-mediated endocytosis. And I'll explain all that. So phagocytosis, cell eating, that's our Pac-Man cell. So essentially what will happen is the cell will extend these pseudopods. That's kind of like what we see here. These pseudopods are shooting out from the cell on either side of this large particle. And it surrounds the particle, and it puts it like in an envelope, much like you put a letter into an envelope, and it pulls it in. So it internalizes that sac, and then fuse with the lysosome. You guys remember what lysosomes are? Those are those digestion organelles. They do a lot, right? But they have these digestive enzymes on the inside, okay? And they'll suck it in, and then they'll fuse those digestive enzymes into that little membrane sac, and it'll start to break down the contents there. Okay? So that's what we see with our white blood cells, like that macrophage. So take a look at this picture here. All right? Phagocytosis. All right? You can see here that the cell, there's a large particle outside the cell. The cell wants it, so it extends these pseudopods around this particle. So they start to wrap around this particle, and eventually it completely encases the particle, and it pinches off, and it forms this vesicle, this vehicle. And then this vesicle will travel in the cytosol, and it'll merge with a lysosome. And then that lysosome will dump its digestive enzymes into the inside, and it breaks this down and digests it. If it's a bacteria or if it's a virus, same thing. It'll dump those digestive enzymes into it and kill it. But it's basically taking something big and breaking it down into its building blocks or its smaller particles there. So that's phagocytosis, cell eating. Now this slide should say penocytosis, okay? But this is what we're seeing. This is the cell drinking. This is we have small fluid droplets outside the cell. Same type of concept. it will start to wrap itself around these fluid droplets and make these vesicles and bring it onto the inside of the cell. The last one here is receptor-mediated endocytosis. We'll see this with lipids, okay? You've heard of good cholesterol, bad cholesterol, HDLs, LDLs, and then we have the very bad cholesterol, the VLDLs, okay? But what we're going to see here is, all right, these types of cells will put these receptors on the outside here. And if you've ever seen videos of bears standing by a river when the salmon are spawning and they're swimming upstream to lay their eggs and whatnot, well, the bear will stand there and the salmon will jump by it and they'll just snag it out of the air. Well, that's what these receptors do. So the cell pops them on the outside of it, and then these receptors, well, as any of those lipids, those HDLs or LDLs, as they're flying by, they'll have a specific, I don't want to call it, well, kind of like a ligand, all right, but not necessarily. They'll have a special structure on the outside, and these receptors will snag the HDL or the LDL, all right? And when it gets enough of them, it starts to wrap itself up, all right? And then it pinches off those receptors, or that part of the plasma membrane with the receptors that have the lipids there, all right? And what it does is then it puts a special, all right, protein coating on the outside of its vesicle. And that protein coating is called clathrin. And that identifies the vesicle as having these lipids on the inside. And it just tells the cell, hey, I got some lipids here for you. All right, let's start our lipid digestion. We'll bring some peroxisomes in maybe, and we'll start to undergo some beta oxidation. Okay? So that's the last one, the receptor-mediated endocytosis. All right, folks, that was the last part for the plasma membrane. So we got 2 down and 1 to go. Okay, so we've covered the cytoplasm and we've covered the plasma membrane. Now we're going to finish up here. Trust me, the nucleus is, there's only 3 slides here. All right, excuse me, 4 slides. Okay, so a couple things about the nucleus. All right, this is the cell's control center. All right, but it's the largest visible structure when you're looking at a cell underneath a microscope. Okay, now some cells, well, excuse me, most cells only have 1 nucleus. Some cells have more than 1, and some cells have less than, or have none, excuse me, less than none would be none. So we've already seen when we were doing the tissues that skeletal cells, or muscle cells, they're multinucleated. Cardiac muscle cells can have 1 to 2 nuclei. All right, and then some cells have none. All right, that would be like a red blood cell, an erythrocyte. Okay. When you were studying the tissues for lab, all right, and we were learning about columnar epithelium, for example, you saw that a columnar epithelium was longer or taller than it is wide. Well, the nucleus will be similar to that. It will be like an oval shape, like an egg. All right, and then when we were talking about squamous epithelium, it's a flattened cell. Well, the nucleus for squamous is going to be flattened as well. So the nucleus will normally mirror the shape of the cell. And I bring up the nucleus because it's one of the most important structures, as we know, because it's the control center, but really it stores this, DNA, our genetic material. It's what makes us us, or who you are. It's the blueprint for everything about you, the color of your eyes, the color of your skin, the color of your hair, how tall you are, how short you are. All right? So pretty much all different characteristics, how well your vision is. Okay? All that information is stored in the DNA inside your nucleus. So much like a cell, all right, your nucleus has its own envelope or limiting barrier. Okay? And we call that the nuclear envelope. So it also has a phospholipid bilayer. Okay? And it also has on the inside of that nuclear envelope some gel, some goo. We call that nucleoplasm, much like the cytoplasm that your cell has. Okay? Well, there's a bunch of holes in your nuclear envelope, and that's to allow movement of certain things into the nucleus and out of the nucleus, because your ribosomes are made inside another structure that lives inside your nucleus, and it has to get the ribosomes out into the cytoplasm. Okay, so you can start making proteins. Okay, so it also allows for certain structures to enter into the nucleus, maybe some ions. All right, or water. We always like water. We need water for almost everything. Okay, so those nuclear pores allow for the movements of things into and out of the nucleus here. And you can see here you have your phospholipid bilayer of our envelope with a bunch of holes here. Those are the pores, the movement of things in and out. This kind of grayish material here, that's your genetic material there, okay, your chromatin. And on the inside of our nucleus, we got a smaller structure, and that's the nucleolus. That's where your ribosomes are made. Okay, so your ribosomes will come out of the nucleolus, remain here, all right, inside the nucleoplasm, but many will leave out through these nuclear pores here. Some will attach themselves onto the endoplasmic reticulum, some will free float, all right, into the cytoplasm. Okay, so finally, the last slide for this video is cell division. Cells like to divide. And what that means is where you had one cell, they'll split and form two cells. Okay? And this is important for damaged cells, all right? Or if a cell gets too old, if you're growing still, when tissue growing like bones, skeletal muscle, we'll see cellular division occur. So there's two types. We'll start with the easier one first. The first type is meiosis. And meiosis is when your sex cells divide. So what are those? That would be the oocytes, or the eggs in females, and then the sperm or spermatozoa in males. Okay? And mitosis is cell division for all other cells in your body. So that's how I describe it to folks. Meiosis is the sex cells, mitosis is everything else, and those cells are called somatic cells. And when I say everything else for the cells that are able to divide, because there's some cells that do divide. Neurons, they do not divide. So they're what we refer to as amitotic. And that concludes chapter 4 with an audio recording for you guys. All right, I will try to get this out to you as soon as possible. If you watch this before 7 p.m. on Sunday, September 13th, you will know that it's 3 o'clock now, so we will be hearing each other, hopefully, in about 4 hours. You have a mandatory Blackboard Collaborate session as a graded assignment at 7 p.m. I hope to see your names there. I won't be able to see you. All right. I hope this helped. And if there are any questions, of course, always email me. All righty. Enjoy.